The central dogma of molecular biology tells us that a gene gets transcribed to RNA, which gets processed to messenger RNA, which gets used to make proteins by ribosomes in a process called translation. A lot of times when you think about how much a protein is being expressed or kind of made, people focus on the level of transcription. So how much of those messenger RNA copies are being made? And you can measure that with things like RNA sequencing, but that's only one piece of the puzzle. You also have to consider translation, as well as post-translational things like the protein actually getting degraded, which I'm not going to go into here. I want to address this translation aspect because there are specific techniques that scientists can use, such as polysome profiling and ribosome sequencing, also known as ribosome footprinting or ribosome profiling. These methods can tell you kind of how much a messenger RNA transcript is being used, as well as more specific things in the case of like your Ribo-Seq about where the ribosome is actually located. And you can also get even more detailed information with in vitro techniques like Toeprinting, inverse Toeprinting. So I will go into all of these techniques in a little bit of detail. I have practiced with ribosome profiling, lots of practice, and I'll tell you a little bit more about my recent work. So let's dive in. The most common ones you're going to see are going to be your ribosome footprinting and your polysome profiling. So it's a little confusing because ribosome footprinting has like a bunch of different names. It's also known as ribosome profiling or Ribo-Seq or ribosome sequencing. The basic idea here is that you're trying to figure out where along transcripts the ribosomes are. And sometimes you want to know this because you want to know like where exactly the ribosomes are. Like, are they getting held up at specific sequences? And that's kind of what my research was focusing on. But you can also use it just because it's a way that you can use sequencing in order to figure out how many ribosomes there were per transcript. The way that this works is basically you allow those ribosomes to do their translating, then you chew up the RNA around them. They are going to be protecting the RNA that they're sitting directly on. So if you can then isolate that RNA and sequence it, well, then you can figure out where the ribosomes were. If you align those sequences to the genome, then you're able to kind of see if there's a pileup, see how many ribosomes there were on a transcript, all that cool stuff. And you get a kind of more unbiased view of things. And if you want to get a measure of kind of overall translation efficiency, so that would be like how many ribosomes there are per transcript, you're also going to need to know how many copies of the transcript there are. And so then you can combine it with total RNA sequencing in order to get that information. Thankfully, total RNA sequencing is way easier. There are kits that make it super simple. And you can combine this with your ribosome profiling and to calculate that like translational efficiency. But remembering that that can be a bit misleading, similarly to with polysome profiling, because you could have a lot of ribosomes that are kind of built up and not actually productively translating. And that would still show up as a high translational efficiency. That'll actually give you specifics about the transcript, but there's also a method called polysome profiling, where here you basically are going to separate the polysomes from the monosomes and further granulate the polysomes and things. Basically, what am I talking about? Messenger RNAs, they get used by ribosomes, but it's not like one ribosome uses one messenger RNA at a time. There's plenty of room on that RNA for multiple ribosomes to get involved. And often they do. So if a gene is being highly used, a transcript is being highly used, highly translated, it's going to have a lot of ribosomes on it. And we call this a polysome. Polysome profiling is going to take advantage of the fact that when you have a lot of ribosomes on the transcript, it's going to be heavier and it's going to sink further in a sucrose gradient. And then you can go, and if you're interested in a specific messenger RNA transcript, you can do things like qPCR or Northern blot to see if it was in that fraction. You can also just kind of look at global changes in whether the cells are like translating more or translating less under various conditions. Are things getting held up? So that's one of the kind of downsides of a polysome profiling is because you're not seeing where the ribosomes are. You can't tell if there's a lot of ribosomes on a transcript because it's just like they're all held up at some point, or it's because it's just being used a lot. So ribosome sequencing can get you that. So polysome profiling is really good about telling you how many ribosomes are on the mRNA, whereas ribosome footprinting is really good for telling you where on the mRNA those ribosomes are. So if you kind of think about, and it's a small world ride, And imagine there are lots of copies of this It's a Small World ride. If you want to know how well, like how much traffic there is on the ride. So imagine if the boats only go if there's a full crew of people. So the more people that want to go on It's a Small World, the more of these boats you're going to have per ride. And then you have lots of copies of these rides. Polysome profiling would be kind of telling you how many boats were on the ride. Whereas ribosome profiling would be telling you, like, if those boats were held up at a specific place along the route, that ribosome profiling would allow you to see those holdups. So maybe there was some sort of barrier in the path of the boat, and so they were all getting tied up there. So I'll go more into detail about each of those techniques in a minute and what they're good for. But then I'm going to tell you a little bit about Toeprinting and inverse Toeprinting. So I just thought I'd give a quick few words about them here. Those are methods you can use if you have a known sequence that you want to analyze the ribosome's position on. So maybe you want to verify or validate the kind of initiation start or termination state. Or maybe you want to see whether a various protein is important for initiation or translation, or termination. And so you're kind of experimenting with different things in your mixture. Bottom line is you take one of these transcripts and in Toeprinting, you're going to get a ribosome to translate it, and then you're going to stop the ribosome and use a reverse transcriptase in order to kind of go and make from the 3' end, go backwards and kind of make the cDNA until it hits the ribosome. Then what's going to happen is that if you had that labeled at the end, you're going to get a piece of the cDNA that then will correspond to the length of the transcript. And then you can run this big sequencing gel in order to kind of see where things were. That requires the sequencing gel. Inverse Toeprinting does not. It uses sequencing. So just like Illumina, that sort of thing, kind of like Ribo-Seq. What it does here is basically it's going to go from the opposite end. It's going to use an exoribonuclease to chew up the ribosome. So instead of making cDNA, so instead of like reverse transcribing from the 3' end, it's going to actually like chew up from the 3' end. Then you're going to be left with the 5' end that you can then sequence and get the location of the ribosome. But those are kind of more obscure methods, more specialized methods. Ribosome footprinting or polysome profiling are the ones that you more typically come about. So let me start there. Polysome profiling. So again, the idea here is that a monosome is when you have a single ribosome on a transcript, and that's not very efficient if you want to make a lot of protein. The amount of protein that you can make is determined, remember, by how many transcripts you make, so transcription, and then how long those transcripts stay around, so post-transcriptional regulation, and then how much translation goes on, so this translational regulation and things. Messenger RNAs that are used a lot are going to have a lot of ribosomes on them. And so when we have a lot of ribosomes on a transcript, we call it a polysome. Now, polysome profiling takes advantage of this thing called sucrose gradient sedimentation. So a sucrose gradient is basically you make a gradient of sucrose, good old sugar. You make this sugar mixture and you can use, there are various ways you can do it without a machine, but this machine has a nice gradient maker. You can't tell here, but this is like a little platform that actually moves and rotates and things. And so if you stick a solution that has kind of like a stepwise gradient, and then you have it turn on its edge and then you spin it, you'll get this even gradient. And then you can use this gradient in an ultracentrifuge with a swinging bucket rotor in order to spin the molecules, and then they'll separate by size. And so with the polysome sedimentation gradient, what you're going to do here is you're going to take all those transcripts that have the ribosomes on them, and you're going to spin them down in that manner. The more ribosomes they have on them, the more they'll sink. And then you go and you see where they end up. And so you can see where they end up because RNA is going to absorb UV light. And so this part of the machine, this is a biocomp, it's really cool. This thing here, it's actually going to go down in the tube. So you take those tubes out, your sucrose gradient out of the very, 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 very carefully out of the ultracentrifuge, and then you're going to stick it in here. And there's a little adapter thing, and then this goes down and it kind of pushes down. It has this plunger on the top. It's actually going to pull the liquid out of the top, and it's going to go into this through a UV detector and into a fraction collector. And that way you're able to kind of see where the ribosome, the ribosomes are. Then what you can do is you're going to get some sort of like curve like this. And if you collect fractions of these, you can then go and you can use like qPCR, you can use a northern blot, you can use things like that in order to look for specific transcripts, or you can just kind of get a general idea of what's going on with translation. Like, are you getting more polysomes? Are you getting less polysomes? If you're getting a lot of monosomes, that might mean that you're having some problems with translation. So when it comes to interpreting a polysome profiling, so you're going to be seeing one of these like UV traces that's coming from looking at the sample that was taken from the gradient. On your left is like the light stuff, and on the right is on your heavy stuff. So if you think about what's lightest, that would be like free mRNA, other RNAs that are light, so they're not thinking, but they're also absorbing that light. So not all of it's going to be actually your ribosomes. There could be other, not all of it's going to be like your mRNA. There could be other things as well. And those will all kind of be in this shoulder part. Then you're going to get the individual ribosome subunits. So depending on what ribosome species you're looking at, these subunits will have different sizes, but you'll get your small subunit, your large subunit. Then you'll get your monosomes, which are typically like the poorly translated messenger RNAs, and then the polysomes, so the well-translated ones. So you can kind of, for the first few ribosomes, be able to tell if there are multiple on them, like 2 or 3 or 4 or 5, but then when there's so many, you can't really tell, and so you get kind of like this big peak. Well, you get a big peak if you have a lot of ribosomes on individual transcripts. If you have fewer mRNAs on multiple that have multiple ribosomes on them, you're going to see a shift in basically having more of those monosomes and less of the polysomes. For example, if you disrupt your global translation with arsenic, you'd see a shift like that. You can also then, if you wanted to, like look at individual fractions. So basically, when you're collecting them from their gradient collector thing, split it up into different fractions and take a look at what's in each using something like your qPCR, like your northern blot, all that good stuff, and things like this. So kind of because you're not typically sequencing all of these, it's kind of like the difference between doing like qPCR versus RNA-seq. And when you think about like ribo-seq versus polysome profiling, polysome profiling is not going to tell you the exact sequences of everything that's there. But if you know the sequence to look for, you can go and you can see if it's there. Similarly, with like qPCR, if you know what transcript to look for, you can go and measure the amount of that transcript. But if you don't know what to look for, you're not going to see it. But if you do RNA sequencing, you will. But sequencing is a lot more work. And so it makes sense that if you are interested in a specific transcript, you might want to go the polysome sedimentation route, as well as kind of just getting a global look at translation. But let's go back to ribosome footprinting. So again, this is called like RiboSeq or ribosome sequencing as well. And basically the idea here is that you're going to take ribosomes that are translating a transcript. You're going to use RNases, so RNA chewers, to actually chew up around the ribosomes. And so it's a bit of a troubleshooting and optimizing act to try to get to a point where you're chewing up just around the ribosome so that all you get sequence-wise left over is going to be the place that the ribosomes were sitting on, without actually just like chewing up the ribosomes, because remember, ribosomes are mostly RNA as well. The basic idea here is that, well, the point that the ribosome is sitting on is going to be protected from those RNases. And so if you can go and you can sequence those RNAs that it was sitting on, you can see where the ribosomes were located. You can see where they were translating. You can see maybe if there was a holdup somewhere. So sometimes there can be holdups for things like there's a region that has some weird secondary structure. It could be that there's some protein that's sitting there and blocking translation. It could be like a rare codon, structural barrier, various things like that. You could also, there's often like a buildup at the initiation sites. So all of those sorts of things can be shown in RiboSeq, because what you get is kind of like the average of like kind of like the combined position of all the ribosomes, because you're going to get the sequence of each of these little pieces, and then you're going to align those to a genome. And so if they had more ribosomes at that piece, you're going to sequence that piece more, and it's going to show up more on your curve. The actual process of doing ribosome profiling can be a really, really long process. So there are some simpler methods, but kind of this is the method that I was using, this kind of like more classic method. A lot of times the simpler methods might not be quite as accurate and things like this. But this method a lot of different steps. So basically you take the cells and you break them open. So there are different ways. You don't want the ribosomes to just like keep translating and translate all the way off if you're trying to figure out like what was happening in the cell or in the mixture at the time. So often people will like add antibiotics to stop the translation, or do like a flash freezing, do some like I did like fast filtration freezing, because antibiotics can cause like artifacts, causing the ribosomes to stall at specific sites. But you're going to lyse things open, you're going to lyse the cells open, you're going to digest with your RNAs and isolate your monosomes. There are different ways to do this. So you can use like a little gel sec column, like this little kind of like spin column for isolating the cytoplasmic ribosomes, or I was actually working with mitochondrial ribosomes. And for that, I did a sucrose gradient and then separated them that way. There are also various like antibody-based methods to pull out the ribosomes. You then isolate the footprints, do an RNA extraction. I did like an RNA cleanup, and then a size selection polyacrylamide gel electrophoresis. So basically, you're going to run a gel, cut out the band of the size that you're interested in, put linkers on it, reverse transcribe it, purify that DNA, circularize it, purify that, and then there's a bunch of like sequence quantifying it, making a library. So when we talk about a library, that's just like a large collection of molecules. So when it comes to like a sequencing library, that's basically just the genetic contents of a sample split up into fragments of DNA prepared for sequencing. And when we talk about preparing for sequencing, well, this involves like putting adapters on the end that make it so that things like Illumina sequencing, they can bind to the chips, and then they can be sequence to figure out what sequences that the DNA was, which corresponds to what the sequences of the RNA was, which corresponds to what was the position of the ribosomes. Then you can do basically align them to the genome in order to figure out where the ribosomes were. So I did this with mitochondrial ribosome profiling, and so we got some really cool results. And so if you want to go check them out there, feel free to check out blueprints. Yes, mitochondria have their own ribosomes. They're more complicated to work with, but they're really cool. But what's not so cool is that they can actually get inhibited by antibiotics that are meant to target bacterial ribosomes, because mitochondrial ribosomes are similar to bacterial ribosomes in like their core conserved region. One of the interesting things that we found was that, similar to the case in bacteria, some antibiotics, so we tested chloramphenicol and linezolid, they actually cause the ribosome to stall at specific sequences. Most particularly when there's like an alanine in the -1 site of the nascent chain, in position to the P site, so like the penultimate residue in the nascent chain. We're able to figure this out by doing this mitoribosome profiling, and then doing like logo analysis to see which amino acids were enriched. Now, ribosome profiling, it can kind of give you the position of the ribosomes, but if you want to be like super duper accurate, if you want to make sure, like ensure that it's actually stopping at this position, maybe you want to see if the position around it changes, it makes some changes and see if it still gets inhibited, all that good stuff, you're going to need to turn to some sort of in vitro system where you can manipulate the transcripts and then measure kind of where the ribosome's stopping. This works if you have like an in vitro translation system, which thankfully there are some really, really good ones for bacteria. So hopefully in the future, people will be able to do some Toeprinting and kind of like follow up on the results. But the problem is mitochondria have really complicated translation systems, and so it's not as easy to study in vitro translation in the mitochondrial system, although people are making some really great work with it, including some of our collaborators, the Nono Takeuchi Tomita lab. So going back though, what exactly is this Toeprinting and inverse Toeprinting? So say we found a sequence that we were interested in based on the ribosome profiling. What we would then do would be to take that sequence and take a part of that sequence and actually just stick it in an in vitro translation system, give it some ribosomes, give it all the stuff it needs to translate, and then let it translate. And then we would add the drug or do whatever and see that would stop the ribosome. We would then use a reverse transcriptase to create with a labeled primer. So basically like radioactively labeled often, or you can have like a fluorescently labeled. And what it's going to do is then the reverse transcriptase will start there, and it will start reverse transcribing at the 3' end of the transcript towards the ribosome, and then it's going to stop when it hits the ribosome. So then you're going to have a piece that's labeled, and then basically, depending on how far where the ribosome was positioned, the piece is going to be longer, the piece is going to be shorter, and then you go and you look and see where that piece is compared to like a letter labeled template. Inverse toe printing, you go from the other end, and so you chew up instead of making a copy of that 3' end of the transcript, and then you use a DNA polymerase to make cDNA from the 5' end to the ribosome's position, instead of going from the other direction. And instead of running a gel, you're then going to sequence it. One of the useful things about these two, in addition to just finding stall sites, is to identify things like open reading frames. Ribosome sequencing can also be used for this. And so basically an open reading frame, that's the part that starts with like a start codon, and then the ribosome translates along it until it reaches a stop codon. That is an open reading frame. Typically there's a main one that's going to be sometimes it's called the ACORF, the annotated canonical ORF. And basically this is where you get the normal protein. And so it's typically starting at an AUG and you make this normal protein. But there can also be upstream ORFs. And so basically these would be located in the 5' UTR. And there's some different options depending on what's happening, where they're positioned, what's in between them and the regular transcript. So if they're in a different frame or there's a stop codon between them and the regular transcript, you could either end up getting like a little peptide, or you might get a different protein that's like really different, might be functional, maybe degraded. It all depends. If it's in the same frame, though, as your main or your annotated open reading frame, and there's not a stop codon between them, what's going to happen is you'll get a protein that has a longer end terminus. Sometimes these can be functional, sometimes these can have cool functions, sometimes it might just mess up your protein. These upstream ORS may also be regulated by the normal ACRS, and vice versa. So you can have translation from one of these upstream ORFs that's going to basically interfere with translation from your normal one, such as by hiding the ribosomes or the ribosome binding sites, kind of like stalling the ribosomes, things like this. Or it might be that translation at that upstream site is kind of going to unwind some secondary structure that was holding things up. And you can also get like reinitiation at the normal ORF if there's like favorable conditions, if you start translating at an upstream ORF. So there's a lot of work that's going into this sort of thing. And one of the things is that like ribosome sequencing, sometimes like it might give you like false like impression that there is an upstream ORF when there really isn't. And so then often people might follow up with something like toe printing or inverse toe printing in order to kind of figure out what's going on. Toe printing and inverse toe printing, not only can they like validate the ribosome is actually starting there or stopping there and things like this, but you can also, because it's in vitro, you can manipulate the sequence in order to see sequence determinants of whatever event you're looking at. So a little bit more about toe printing. So sometimes this is also called primer extension inhibition. And so you take that cDNA that is complementary to the 3' end of the transcript, and you have this be labeled. So typically it's going to be radioactively labeled. So typically you're going to label this cDNA, the end of it, with your P32 ATP. And then what you're going to do is you're going to allow the reverse transcriptase to transcribe until it hits the ribosome. You're then going to purify out the cDNA, run a sequencing gel, and combine the size of this fragment to the two known positions from a template in which you kind of like labeled the different letters one at a time. So you'd have like one where you use radioactive A's, one where you use radioactive C's, one where you use radioactive T's, and one where you use radioactive G's, and then you like make cuts and stuff like this. So it can be a lot of work. So there are like methods. So I say use the sequencing gel, but there are also newer methods where it can kind of use fluorescently labeled primers and a sequencing machine for the readout. So hopefully this will become more widestream or mainstream in the future. But for now, like the kind of classic way is to run one of these gels. One of the kind of confusing things about this is that because the ribosome is kind of going to be blocking part of things, the size you see is going to correspond to the 3' end of the ribosome, not like the part that the ribosome was actually like this part of the sequence that the ribosome was actually like working on right then, but the part that it was going to work on next. And so the piece is going to be a little shorter, and so that can kind of confuse how you're reading it. because the stop position of the cDNA is, depending on the species, going to be like 13 or 18 nucleotide downstream of the P position because the ribosome protects what it sits on. Okay, so you're going to see some gel like this, and hopefully the authors have kind of like annotated the side so that you can tell what corresponds to what. If you look up, what kind of things can you see? Well, in the 5' UTR region, you might be able to detect initiation on an upstream open reading frame, but most of them should be at this like canonical start site. So it's typically going to be your ATG or your AUG in the RNA, but this is going to be the cDNA that you're looking at, so that would be a T. Within the open reading frame, you can detect stalls. So if you see like this strong band within the open reading frame, that would indicate that a ribosome is stalling at a position. You can also detect like termination sites because termination is slow. So you can see the termination sites and might be able to see premature termination. And then again, the signal you'll see will be downstream of the actual stall point. So figures will typically be annotated to reflect this. So here, the ribosome, you might see it band here, but that's actually corresponding to the stop because the ribosome is taking up this extra position. So often you're going to see that's the sidebar is going to be annotated to adjust that and take into account the extra bulk of the ribosome. So to get if you're looking for like an initiation site, or typically you want to do this anyway, just to kind of get the start site, you can use a drug that actually that inhibits elongation but not initiation, that leads to a buildup at start codons, allowing you to detect the initiation site. So you might see something like a lane where they use cyclohexamide in order to stop elongation, and then you're going to see the position of the ribosomes at the start sites, including potentially at those upstream start sites or other start sites. Stall sites, these can be anywhere, just wherever the ribosome is stalling. And they can be induced in some cases by specific drugs, such as the example I was talking about, or metabolites. And so these can actually be used, like in bacteria, in order to kind of determine when to translate specific sequences. They can also use like drugs to try to antibiotics can actually induce the stalling in a way that's going to make it so that they translate things. So lots of cool stuff like that, going back to kind of the discussion of open reading frames. You can also detect termination. So ribosomes are going to stall at termination sites because it's slow. So you're going to be able to detect that. And so, yeah, so bigger things up top, smaller things down low. Magnesium, this is going to inhibit translation at all translations. This is your negative control. You get the full-length cDNA because there's no ribosome blocking the reverse transcriptase. When you have an elongation inhibitor, you're going to see the initiation sites. If you have a stall inducer, you're going to see stalls where at that point. You always should see the termination sites because those are going to be kind of just a slow point, and then any inherent stall points are going to be just show up. As for inverse Toeprinting, well, here you're just going from the other end and you're using sequencing. One of the benefits about this is you can kind of scale it up and use like a large library of sequences to translate. And in that way, you're able to test a larger range because Toeprinting is pretty low throughput. So here's a cool example that kind of set the impetus for our project. So Marx et al. Context-specific inhibition and translation by ribosomal antibiotics targeting the peptidyl transfer center. So here they found that some of the antibiotics arrest the bacterial ribosome at specific sequences. So they also tested chloramphenicol and linezolid. And you can see in this paper that they use Toeprinting of kind of known sequences that bacteria are known to stall at. And they use these gels, this ribosome Toeprinting experiment, in order to kind of analyze the ribosome's position, where it's stalled on those positions. So you can see, like when you add chloramphenicol, the drug induces stalls at specific sites. They also did ribosome profiling. And so with ribosome profiling, you get something like this, where you get the ribosome density, so like reads per million at specific sequences, and then you're able to, those would be mapped to the genome, and you can then go and look at them. And so you can see like a redistribution, and you can see a pileup at specific sequences. You can then go and you can analyze more in detail the composition of those sequences, like were there specific amino acids that were overrepresented, were there specific codons overrepresented, and things like this. So lots of cool stuff, and we found very similar things in mitochondria with some differences. So lots of cool stuff there, and the potential for designing antibiotics that maybe have less off-target effects on mitochondrial ribosomes. So check that out if you're interested in, but do check this article as well. There are also methods where you can use to just like purify ribosomes. So we did some structural work, and basically to purify ribosomes, you're going to take advantage of some sucrose cushions and sucrose gradients. So sucrose cushions are going to basically be a bulk separation, whereas, and you're kind of gonna, because the ribosomes are so heavy, they're just kind of sediment and get stuck in that, like under the sucrose. And then you can remove the extra stuff. Oh my God, sucrose cushions. The ribosomes get so like stuck, like such a hard pellet that can be really, really hard to resuspend. But once you resuspend it, then you can stick it in one of those sucrose gradients where you can separate the complete ribosomes from like individual subunits and do things like this. Then, depending on what kind of ribosome and things like this, you might precipitate it and resuspend it in a buffer you want, or you might do some buffer exchange with a different method. So ribosomes, super duper cool, and lots of methods to study them and lots of methods to study translation based on them. So to review, we've got like our ribosome footprinting, also known as ribosome profiling or ribo-Seq, where you go and you look at where the ribosomes are sitting. And the way that you do this is by chewing up the RNA around the ribosomes, isolating the portions that they were sitting on, making cDNA from them, and then sequencing it, aligning it to a genome, and seeing where the ribosomes were bound. With polysome profiling, here you're going to take advantage of the fact that highly translated transcripts are going to have more ribosomes on them. We call those transcripts with lots of ribosomes on them polysomes. They're going to be heavy, they're going to sink in a sucrose gradient, and if you can go and look and see how many ribosomes were on a transcript that you're interested in, taking advantage of their sinkingness and the UV detection of the ribosomes. And there are also more specialized techniques if you want to go and look exactly where the ribosome is. You want to do some manipulations to the sequence to see what parts of it are important. You want to test the importance of various components of the translation mixture. Toeprinting and inverse toeprinting are going to be good for this. With Toeprinting, you are making a cDNA copy of the 3' end of the transcript to the position the ribosome is stuck at when you stop, and then you're going to run a gel to see what that position, how long the piece is and what it corresponds to in the sequence. Inverse Toeprinting, you're going the other direction, you're chewing up that RNA instead of copying it, then you're going to make a copy from the other end and sequence it to see where the ribosome was, and you on that transcript getting to like the position of its 3' end.